let me take you 25 years back in time. I must have been around 1.5 when I first came across origami. And it looks like I found it really fascinating how my mum can make a boat from just a piece of paper. Back then, I had no idea what real artists can make from a single piece of paper without scissors or glue. And I had no idea that origami would become the topic of my PhD in physics. Today, I'm not folding paper, I am folding DNA. DNA origami. And I don't mean folding the DNA double helix from paper, even though it looks really beautiful. I literally mean folding the molecule. I am folding small channels or pipes from DNA origami, hoping that one day they might even save a life. And now, I guess I owe you the answer to 2 questions. First, how do you fold DNA? And second, why would you make channels? So, how do you fold DNA? Well, nature uses DNA to store information, the information of life. Volumes of history are written in the ancient genetic alphabet of A and T, C and G, stories of the present and the past that we're just starting to decipher. And while we are still deciphering these stories, we scientists have also started to use DNA as a construction material. Thanks to the groundwork of Ned Seaman and Paul Rothman, we are folding DNA into nanometer-tall objects. So we're essentially building with the building blocks of nature. No genetics, pure architecture. And just like paper origami follows simple rules, you fold and you turn a piece of paper, DNA origami also follows simple rules. And the simple rule for DNA origami is the so-called base pairing of DNA. The DNA double helix is made from 2 strands, so we could break them apart in the middle. And if the DNA sequence, the genetic alphabet of these 2 strands matches, they bind and form the double helix, which we know. But if they don't match, they don't bind. And that is the simple rule that governs DNA folding. Imagine you had a long single strand of DNA with a given sequence. You would add some short pieces which match, bind in specific places, form a double helix. Now we've got something like a loop. And with many of these short pieces, you could fold the long piece up into any shape, crane, or with different ones, you could fold a crocodile. In practice, you just go online, you download a program which is available for free, you literally draw the shape you want, the program calculates the DNA sequences you need, you email them off to a company which makes DNA, you mix the DNA with a pipette, you heat it up, cool it down slowly, and after a few hours you've got billions and billions of crocodiles in just a droplet of water. You don't believe me? Well, you can take the crocodiles to a special microscope, the atomic force microscope, and look at them. And you can imagine it really made our day when we found these 2 little guys playing. And of course, no crocodiles were harmed in the making of this presentation, because all you see here is DNA. So these are DNA-shaped objects in the shape of a crocodile. Why a crocodile? Well, the brick crocodile is the logo of the Cavendish Laboratory in Cambridge, UK, where I work today, and more importantly, where Watson and Crick discovered the structure of DNA. And I'm not sure they did anticipate that one day there'd be a DNA version of that logo. So we can make crocodiles, but if we can make crocodiles, we can probably make anything else, right? We could, for instance, make channels. And that brings me back to my second question. Why would you make channels? Well, channels exist in every single one of the cells in our body. They sit in the envelope, in the membrane that surrounds the cell, and they are vital because they enable communication. And we all know that communication is vital. I mean, we can't survive a day without our smartphones, right? Without communication, our civilization would break down immediately. So I honor the people who study communication, linguists, psychologists, and those who facilitate it through mediation or through technology. But just like communication is vital for us, it is vital for ourselves. Our cells need to communicate with each other. They need to gather information about their environment. They need to transmit and receive signals. And they can do all this because they have got thousands of channels in their membrane. I like to call them the cell phones of the cell because they literally are the communication devices of cells. And we all know what happens when communication goes wrong on the human level, right? We make a mistake, we lose a friend, or we even start a war. And unfortunately, if communication goes wrong on the level of cells, it is not less lethal. We get diseases like cystic fibrosis, Alzheimer, cancer, schizophrenia, the list is endless. So we need to understand communication. But what I cannot create, I do not understand. The famous physicist Feynman once said. And following this maxim, we are creating artificial channels to understand the natural ones and so that in the future we can hopefully design better drugs or replace defective channels in cells. In fact, 50% of the drugs that we use today target natural channels in cells. So imagine what we could do if we could create artificial channels exactly the way we need them. But to make such channels, we'd need to be able to build on the nanoscale, right? We'd need to be able to make billions of copies of identical channels just like nature does. And we should use a material that is cheap, biodegradable, safe to use, available everywhere. Sounds like a challenge, but luckily we have a technique that fulfills all these criteria. DNA origami. And now we are back. So we can use DNA origami to make crocodiles, or we could use it to make channels, right? DNA is a great material, probably the greatest invention of nature. I mean, it's really stable. We can still recover the DNA from 10,000-year-old horse bones, and we can really predict and design its folding. We can make crocodiles, or we can make channels. They could look like this one. I think it looks a bit like it could really be the future megaphone of a cell. This is again how it looks under the atomic force microscope. And of course we can make little brothers or sisters as well, inspired by the diversity of natural membrane components limited by our imagination, all made just using DNA. But now, how do we go from channel-shaped objects, from shape, to function? How do we get these channels to self-assemble in the membrane of a cell? Well, we can attach molecules to DNA, and we could, for instance, attach lipid molecules, which could hopefully anchor these channels in a membrane. And I'm glad, I like to thank all the other people who've been working on this, because I think that proves that the idea is not totally crazy, that this could actually work. But how do we prove it works? As scientists, we need to prove it works, right? My friend made this paper origami model of a channel for me. And if this model was the size of the actual DNA origami channel, a grain of sand would be larger than Mount Everest. So these channels are tiny. How do we know they actually punch a hole in the membrane if they are way too small to see? Well, it's great we're using DNA because we can attach fluorescent dyes to DNA, molecules which glow if you shine light on them, if you shine a laser on them. So we can make these channels glow, and then we can make membrane vesicles, basically cells without all the mess inside, add the channels, turn on the laser, take them to a microscope, and now I'm showing you real microscope images. So on the left, the channels have not inserted into the membrane, and on the right, the channels are indeed sitting in the membrane. It's obvious, right? But now, how do we know our channels can actually transmit a signal? Again, we can do a little trick. We seal a glass capillary with a membrane, we add the channels, we apply a voltage, and we record the current. Now, if a channel comes and punches a hole into the membrane, the seal is no longer perfect, and we can observe a current going through. If a second channel comes, we see a second jump. So we can really watch these channels do what we designed them to do. So we can use DNA origami to build artificial channels. We can make them react to signals. we can try and mimic the fundamental and fascinating communication that is going on on the level of our cells, in every cell, every day, every minute, and right now. I do admit, today we're still humble apprentices of nature, because nature has made billions of channels, whereas we have just made a few. But one day, we might use these channels to deliver drugs, to kill diseased cells, to stimulate heart cells that beat too slowly, or quite possibly to do something that we can't even imagine right now. Origami may someday even save a life, says paper origami artist Robert Lang. And I think he's right. And I hope the same may be true for DNA origami. Thank you.